Yeah. Hi, everyone. Thanks a lot for coming. I guess this is the first biodiversity seminar of the semester. Please welcome Daniel Wegmann. I'm very happy that he's here today with us at VSL. So for those of you who don't know him, he is a computational biologist, I would say so. A professor at the University of Fribourg, affiliated with the Swiss Institute for Bioinformatics. And so his work is focused on developing statistical and computational methods with a focus on sparse and noisy data, I think. And so, yeah, he's basically has two axes in his research, population genetics and conservation of nature conservation, basically. I'm not sure. I mean, I think your talk will mostly focus on nature conservation, but he's also been working with the ancient DNA, etc. And so, yeah, I'm very excited to hear about your talk, and floor is yours.
Thank you very much. So it's a real pleasure to be here for many reasons. It's the first time for me at VSL, so that's already cool. But also it's the first time ever I was invited to give a talk on our conservation ecology branch. So this is a cool exercise for me, because usually I get excited about these things. So this is human fossils about here 4,000 years old. Okay, and people somehow manage to extract DNA from these bones, and then this DNA has a lot of challenges and issues. That's where we as bioinformaticians come in and try to solve, to kind of elucidate human history. And if you think that looks a bit dull, I can assure you there was a lot of action in human history. So this is actually from a battlefield in northern Germany where nobody knows who was actually fighting, because it's about 1,000 years before the Romans arrived, okay? But today, I'm fortunate that we're going to talk about something else, and that we also started working on, which is really in nature conservation. And for me, this journey started when I was approached by this guy. So this is Thierry Abicher, who wanted to do a PhD with me. And he had a very clear vision of what he wanted to do. He wanted to work in the eastern part of the Central African Republic, in the Chinko area, where he and a friend of his just actually did a private expedition before starting the PhD. And he thought this region had a lot of potential, but also a lot of challenges. And he thought like somebody with more statistical skills would actually be appropriate. I was super skeptical, but excited at the same time. And it worked out extremely well. And today I'm going to show you a little bit about what we do in this region. So first, why did Thierry and his friend Raphael Hickisch actually choose this place? So here's a map of Africa, so two maps. And what you see on the left, I have to figure out how this thing works. So here we see like kind of this band of the northern kind of like savanna belt, just north of the main rainforest. This is a belt that is extremely or extensively used, intensively used by humans. And on the right, we see like the global human footprint index, and you can see that this belt is actually one of the most densely used by humans in Africa. But there is this very peculiar hole right here, where like actually human impact seems to be very low. And that was a reason for Thierry and Raphael to actually say, well, we have to go there. And it turned out that there was extremely little known about this area, because for decades, no scientific publications arose from that particular area. So these two people then kind of embarked on a mission. So this is a very interesting area. This is a picture I took from the little plane that you have to take to actually fly into when I was visiting the first time. And what makes this area, I think, very interesting from an ecological point of view is that it's extremely diverse in habitats. So what you see here is the predominant habitat. Sorry, wrong button here. This is kind of like woodland savanna. But then all along little streams, you see tropical rainforest. And this is true tropical rainforest that harbors up to 90% of the Congolian species, which is really remarkable, I think. I'm not an ecologist, maybe that's why, but I think it's really remarkable because in many parts, this rainforest is just 4, 5, maybe 6 major trees wide. And it just goes all along those little streams. If you're in the ground, in the forest, it looks like this. So it's a actual true rainforest. And you have to walk maybe 5 minutes, and you're in the midst of like woodland savanna. Okay, and so I showed here this picture with some people, so the people working there in the area. So you get some scale. So you see that the trees here are actually much less tall than in the rainforest. In the rainforest, we get 40, sometimes 50-meter high trees. Here, it's more like 20 meters, but the grass grows up to 3-4 meters in summer and then usually burns. So it's like an area that is very, very diverse in its ecology. Nonetheless, it was considered an area of kind of like, say, low species diversity. But when these two people, so Jerry Abishu and Raphael Hickisch, made their private expedition as students, they actually discovered a few amazing things. So they discovered, for instance, this little mongoose, which is the Boussange mongoose, and that has been the first report of this species in more than 20 years. It was believed to be extinct, but it actually occurred in this area. Another discovery was that there's actually African wild dogs. And African wild dogs are like kind of a pet species for conservationists, I would argue, and it was believed that almost every single population was perfectly known. It just happened so that there's actually a very large population in this area nobody knew about. After his PhD, or during his PhD, he continued working there. And I'll just give you two other nice things. So this is the Nyam Nyam parrot. And what's special about this, in this region, Thierry managed to take the first picture of that parrot ever taken in the wild. This is so rare, but it occurs there. And on the right, we discovered a major population of eastern chimpanzees unknown to science. And for me, this was really all remarkable. So I come from this computational side, and I was aware of all those people modeling biodiversity on the world, and we have satellites, and you would think like, yeah, we know what's going on this planet. But then there is this region where these are just 4 examples, but these are all major species in conservation of high importance, where the specialist groups, IUCN, had no clue these were actually there. And where on all maps of kind of worldwide distribution of biodiversity, you would find this region as being of low importance, low kind of biodiversity. And I really started to wonder kind of, how can it be that there are places on this planet about which we know so little about biodiversity? Right, that harbor major species of conservation concern, but nobody knows about it, okay? And I think there's a few reasons why this is the case. So this is an interesting study, I think, done by Raphael Hickisch, so the guy that went with Thierry, and he followed up a PhD with somebody else, and in that PhD, he started to figure out whether there could actually be some biases, or how can we explain where on this planet biodiversity research is actually conducted. And what he made, he plotted this map, which highlights the regions on our planet where there's the biggest bias between biodiversity and biodiversity publications. So there's like areas where there's a lot of biodiversity, but very little research is done. And not surprisingly, this area we work in is like falling right in between. But the crazy thing is, I would argue a third of Africa is actually falling into that area, right? So that means on the African continent, there's a huge bias between how much biodiversity there is and how much we know about. And I invite you to go back to see a lot of those publications where people actually try to estimate or predict biodiversity along the globe. How many data points do they actually have from Africa? Very often, surprisingly few. And I think that is a real problem. And you can see why this is a problem, because in this study, he tried then to see what kind of covariates explain how many biodiversity publications we have in some regions. And here what you see is the number of kind of like publications from an area correlate positively with the number of protected areas. So the more protected areas you have, the more biodiversity research is done. I mean, I think that's expected. It also kind of slightly correlates positively with the number of predicted terrestrial mammals. So the more mammals are there, the more people like to work there. But it actually also correlates with a lot of other things, such as the mean nightlight index. So the more artificial light there is around, the more we do biodiversity research, or the length of the roads. So the more roads there are in an area, the more biodiversity research is kind of done. So it seems biodiversity research is primarily focusing on regions that are probably safe, right? I could show you how the SNF did not allow us to work in this region because it was considered too dangerous, so I was not allowed to send students. So it has to be safe, it has to be accessible, and it has to have minimal infrastructure. I think that is somewhat dangerous because that has a lot of consequences when we model biodiversity. If all our data comes from somewhat disturbed area and we use this data to predict biodiversity in wild areas, it might quite be that we just underestimate the diversity that is there. And for me, that is kind of like, was very interesting. And I was really started to be really interested in asking this question: how can we actually enable informed conservation management in all parts of the world? So in that 1/3 of Africa, where apparently we currently just don't know enough. And it seems clear what we have to fight with. We have to say these are areas where there's probably very little infrastructure and very few roads, right? And then you see all these papers that say, oh, the only way to kind of track lions is to have this super elaborate setup so that you can count the bristles on their skin, so you identify. And you say like, yeah, okay, but we are in an area where that's just not possible. And I think we have to do is we have to try to think, how can we develop tools and methods such that we can do conservation management everywhere, not just where there's roads and electricity. So my goal is to kind of work towards what I call real-time conservation management, okay? Everywhere in the world. We're decades away from it, okay? But here is what I think what we should do. So for me, this is kind of like the basic conservation management loop. So there's biodiversity out there on our planet that we would like to know something about. In order to know something about, we first need to collect some data. It's like a kind of like a tiring process, right? To actually get data. I think many of you collect data out there, you know, there's a lot of work involved, okay? And once we have data, we want to analyze the data so we get an understanding of the biodiversity out there. And if we have a proper understanding, we can actually propose some conservation action, which then hopefully has a positive feedback back on the biodiversity that we have there. And so this is what people do. And if you read papers from my group, from other groups, what you often notice is that there is a huge gap between when the sampling was done and the analysis was actually published or the conservation action was taken. So in our case, it's often years. And I'm not the only one, right? You read these papers about, just recently we had a paper about tigers in India, where the paper was published, I think 1 or 2 years ago, about the decline 10 years ago in Indian tigers. And you really wonder, like, why is there this huge time lag between what we want to study and when we publish it? Or for me, as a computation biologist, I was wondering, how can statistics actually help so this loop does not take years? Okay, so between years and real time is a big gap. For me, the dream would be real time. You know, you have some device out there that takes a picture of a rare species, it automatically goes to my computer, fancy statistic or bell rings. Okay, that would be the dream. We're far from there. But just if we can go from 5 years to 1 year, I think that would already be kind of a major achievement. And I think there's like basically 3 things we strive for. So the first one is we would like to develop what I call simple methods that answer conservation questions with minimal data. I think we are very often, and these models are great, but very often the models used, they are trying to be so complicated and model everything that in many of the remote areas, you will never have the data to make use of them. So how can we develop something where you need as little data as possible, but it answers an important question? The second one is, let's develop methods that relax requirements for standardization and allow for opportunistic sampling. So in this area we work, what is opportunistic sampling? It goes like this. You have ranges somewhere out there, 500 kilometers to the east, okay? And there's an accident or somebody is sick. So the helicopter has to fly and pick somebody up. What Thierry would do, he's now working in the park, would just say, wow, we've never been in this area. Here's a camera trap mounted somewhere, right? That's opportunistic sampling. If you read camera trapping papers, they tell you, you have to have a grid, okay? And on the grid, you do the, but that's just not feasible in most part. Opportunistic sampling is, I have a means of transportation to somewhere that is going anyways, and I make use of it. So I just use this to get the data point. And so I think we need to work or develop methods that are okay with this, okay? Because that is probably how we're gonna get most of the data. And finally, let's work towards automation, but low tech. No electricity means not so easy to charge batteries, okay? There's all these people say, yeah, we fly around with drones. This area is twice the size of Switzerland, and there's one station in the middle where you can charge your batteries. You do not fly around with drones. Okay, well, the Americans do for other reasons, but conservation people do not fly around with drones. So we have to find kind of low-tech solutions, but that are still kind of in an automated way. So for the rest of the talk, I show you 3 examples of what I mean by this, okay? So for 3 different models that we developed, I'm gonna go through some of the mathematical aspects of these models for those interested in. If you're not interested in, snooze out, that's fine. Like we're gonna come back to where I show you that it actually works. So let's start with a very simple thing that I think is very important in conservation, and that is inferring changes in population trends. And so changes in population trends in the region we work are important for two major things. The first one is if suddenly a species tends to have a more negative trend. So for instance, lions are maybe stable and suddenly they start to decrease. That may indicate that there is an undetected threat. We need to figure out what's going on. And on the opposite, a more positive trend may actually confirm the success of an action, right? So you say, our lions are decreasing. We figure out this is the issue, say poaching. We start an anti-poaching mission. If my anti-poaching mission is successful, lions should stop decreasing, decrease less rapidly, or even increase if I was successful in my action. So a lot of the questions our people in Africa have is just, how can you tell me with 1 or 2 years of data whether my train changed? If we need to do an action, wait 10 years to figure out that, oh yeah, the lions are faring better, that's too long. We need to know after 1 or 2 years, do I do something right or not? Okay, so I think it's a very important question. And so we thought about, like, let's try to develop a very simple method for changes in population trends, a method that actually takes minimal amount of data and has power to detect changes of trends if possible. And so here is the method. So basically we model population sizes to kind of like vary in epochs. We always have like exponential growth with a particular growth rate. So here, for instance, a gamma of like 5%. And then at some time, predefined time, the growth rate may change. So here for a negative one. And then it may change here again for a positive one, right? That's the model. And what we're interested in, like our people would say, like T2, that's where I start a conservation action. And the question would be, is this growth rate here different from this growth rate here? That's the question, okay? And so we say, okay, we model this as relative change. So we define by phi here just the basically the population size at time t relative to that at the beginning. So this is why this starts at like 1. As I said, it's exponential change that may vary. And then we have like survey times. So these little t's here are times at which somebody conducted a survey in the area. Okay. So now we can think like, okay, how if we do this survey and we get data, what might actually impact that data? What do we have to figure out, or what do we have to take into our model? So first, let me denote by X here, just the observations. These are counts, right? This could be how many pictures of lions are taken by this camera trap, or how many trans spores did you notice on this piece of the road, something like this, okay? And I have indices I, J, K. So I is the site, K is the survey time, and J is the method. And that's very important for me because people in the field, if you want to have power, use different methods. They put up camera traps, but they have direct sightings. Whenever they drive with a car and they see something, they note it down, right? So I have different types of observation, and I want to pool these. And we're going to say, initially, just let's say these are distributed Poisson discards with some rate. And now this rate shall actually be a function of all the things that I think matter in nature, okay? This is the initial population size, maybe in that region, the change in population size since the beginning, then A, which would be the activity, right? If you have a region, in the region, if you have a region where all animals run around, you're going to get more observations, and in another region, it's maybe quiet. Then delta here will be the detection probability. Okay, maybe I do spore transects on a road, and a road part is sand, so it's very easy to see the spores. But then on a different part of the road, it's rock. So even if the line passes, I won't detect the spore. So local detection will matter. And then finally, this is the effort, S, which we say we know. I know how many camera traps I put up. I know how many kilometers of road I walked. Okay. And then with this, okay, we can actually do a really nice trick. This was first noted by Lincoln Sauer in '97, but it's really sweet. So we're going to condition on the total number of counts. So this is how many total observations you got. And now it turns out that the conditional distribution, so that is what's the probability that I get 5 camera trapping images at this site on this time, condition on the total number of pictures that I got, is multinomial. And the probabilities that you need for multinomials, these P, they're just the normalized rates. So I need the rate at this one sampling point divided by the rates at all other sampling points. And now you can plug in all this, this thing really has a lot of latency. So we can plug in all this stuff up here. And what you should notice is that almost everything cancels out. And you're only left with two things, which is the actual rate of change, which I'm interested in, and the efforts, which are known. So actually, I have a way to estimate change without the need to estimate activity, detection, and anything like that. Why? Why is this working? This is working because we make the assumption that these things are time invariant for the same size and the same method at the same site and the same method. Okay, so here's a graphical illustration of this. Imagine you have like two locations, okay, here location 1 and 2. And I have two observers, but you can also think this of methods, or say observer 1 is Thierry, who knows everything. Observer 2 is me, who has a hard time distinguishing anything from anything, okay? So both of us go out there and we survey. Now, clearly, I cannot compare Thierry's counts with my counts. Imagine I would go in the first year, that would be this, like, or Thierry goes in the first year, then I go in the second year. Of course, every animal will crash. That's just because I failed to detect them, right? So clearly, this is not a comparison that I can make use of. But I can compare, if we do something like this, I take location, Thierry takes location 1, I take location 2. What I assume is, if Thierry is equally good in both years and he sees more lions, it's because there's more lions. I see a fraction of his lions. But if lions really increase, even I should see more lions in the second year. And that, now you can play around and say, how many combinations of things can you make so that it's actually doable? For instance, that would be a really bad experimental design. Thierry goes in location 1 in the first year, I location 2, and we switch locations. That's terrible. Like, there's nothing I can compare that makes sense, right? But there's a lot of experimental setups we can do that actually allow us to only do kind of what I call within comparisons. I'm comparing same methods, same size, same things. And you can go to very detailed. You can say even same weather, right? Maybe the weather matters. Okay, so let's partition our data in comparisons for which I can assume that whatever I compare is independent of like the actual time, so that if I see a change, it's because there's a real change in nature, not because we use a different method or the weather is different or something like this. Oh, I already pressed. Okay, so basically, based on this idea, we implemented an R package, which we call Burp, which implements this. There's a lot of additional things we did. So we implement an appropriate Jeffreys prior for these gamma rates. We also have a stochastic version where we don't necessarily assume the exponential rates to be exactly the same everywhere, but they can vary stochastically. We have also a negative binomial version because you say, oh, Poisson is not good, okay? We can also relax these assumptions of time invariance if you have covariates. So if you, for instance, know something about your method, maybe you used two different camera trap models and one is better than the other and you're not sure, you can use things like this, okay? And here are some simulations just to show you that I think this works really well. So what I show you here is a case where we had a constant population and then at some time it changed to either a positive or a negative trend. And this gamma 2 here on the bottom, this is the new rate after the change, okay? And then here I have the power. So what is the power to actually detect that change with 95% accuracy? And that is, of course, a function of many things. For instance, it's a function of how big the change was. If it was flat or like constant trajectory and then almost constant, power is lower. If it's constant and then there's really a big change, it gets easier to detect. Makes sense. It also depends on how much data I have. So yellow is I have 5 time points before the change and 5 time points after the change. This, like these kind of like stars here, purple, will be 2 time points before and 2 time points after. Blue is 2 before and 1 year after the change, right? So this has an impact on how much power we have. Also, the number of observations. So here, this would be a case with on average 10 observations per species per year. This is a case with on average 50 observations, and you have more power. But the point is, if your setup is somewhat okay, that is, you have some observations, right? You have an actual change that matters. Your power can be quite high, even if you only have like 1 or 2 time points after the change. And this is exactly what we hope for, okay? So now here's some real data application. So this is a timeline of like this region that we work in. Thierry started his PhD in 2013 with me. In 2012, or 11 and 12, he did an excursion with Raphael. And at that time, they already put out camera trap, but did some track counts. The area is interesting because it used to be a big game hunting area. But in 2010, because of the war in Sudan, there were suddenly a lot of cattle herders coming in the area. In 2014, because of civil war in the region, big game hunting kind of crashed, and Thierry managed to convince the big game hunters to actually turn their hunting ground into a national park. And so this is what's called the Area de Conservation de Chinko, ACC. It was established here. They immediately got money from the EU and US funds to actually start ranger patrols as of the early 2016, and since then have enlarged the area they kind of control. Okay, and so as soon as the park started, Thierry did another round during his PhD, another camera trapping session, and then track counts. And then since 2019, they have done track counts annually. Since 22, it's no longer Thierry, but some other people, okay, which is why it's a different color. And now we want to use this data to check whether actually starting to patrol had an impact on the trajectories of the different species we get, okay? And so here are the results. On the left is a major predators, and the right is major prey species. What you should see immediately is that there's many species that are in this upper triangle. What does this mean? This means that we're pretty sure that the initial growth was negative or lower than the second. So we would have this change of going down and up, right? So just for instance, here for the giant eland, these dashed lines are 0. So we're very sure that before the park, the trend was negative. After the park, it was positive. There will be a positive impact of conservation or of this patrolling. That is true for a lot of species, but interestingly, not for all. This bongo species is very interesting. So this is the large antelope that actually hides in the forest. So it was very difficult to poach, which is why we think it actually didn't benefit really from the conservation action. It didn't do that bad before. We see like there's very little data for some of the predators, like 36 or 38 counts of lions in total across the entire time span. We are still 81% sure that the trend or that these species benefited from like actually the park. Spotted hyena seems just not to care at all, right? So leopards was kind of positive in both. So they were already growing, and now maybe they're growing a bit less. We don't really know, okay? But so you see, this is kind of like one example of how I think a simple method, what I call a simple method, actually helps people on the field. They have this messy data, they have different observers, they have different methods in different years. What we do to do this analysis, for instance, for the track counts, we actually break down the road networks in small parts, because I say I cannot compare this piece of road with that piece of road. I consider them separate methods. So we can, with all this partitioning, we can deal with a lot of noise that is in the data. And in the end, only have a method that only estimates what we really want, which is why I think it has power despite all these issues we have. Okay, I switched to a second example, which is about species distributions in space and time. So for camera trapping data currently, most methods actually analyze, or most people that analyze camera trapping data, they actually use occupancy models. And occupancy models, so these, if you're not familiar with them, they're models that actually model presence and absence of species in different parts. And basically, a benefit of this is that they account for what is called imperfect detection. That means a species can actually be present somewhere, but I fail to see it. So just absence of data does not mean absence of species. And because this is confounded, so this detection as well as presence absence, they usually use environmental covariates to model this, okay? So occupancy models have also quite some shortcomings, I think, for this data. So the first one is that there's actually a notion of kind of a discrete patch. So if I put a camera trap out somewhere opportunistically on a tree, what does it actually mean if I say the species is absent? Do I mean the tree or like 100 meters from the tree? For me, it's like this notion that there exists kind of like some space out there where I can say presence absence for it is kind of weird, because if you have fish in a pond, I get it. If you have animals moving around in space and have a camera trap, I'm not really getting it, okay? But then there's the other thing that also I think a simple presence absence matrix actually ignores a lot of the subtle differences we see in population density. So my people down there, if they want to do conservation, They don't only want to know where's the species present in absence. They only want to say which are the most important areas for that species that I need to protect, which is actually going beyond just having presence absence. I want to get some sort of estimate of density. And so we propose a new way of modeling camera trapping data, which we call TOMCAT, which stands for Time-Dependent Observation Model for Camera Trapping Data. It's mostly just because we like TOMCAT as a name, huh? But like, this is what it is. And so here is how the model works. So basically we say, this is the important quantity, this lambda. This is the rate at which my camera trapping device takes pictures. This is kind of like, I think now people in ecology say, what is this? We never worked with this. But I think it's a very natural quantity, which is I go to a specific place in the space, in the forest, in the savannah, I camp there. What is my chance that I'm going to see a lion walking by? Now, this is what this is. This is like, at how long do I have to wait until I see that species at a specific point in time? And then we say that depends on three things. It depends on a relative measure of local density. So that is like, is this a good spot for lions or is this a bad spot for lions? But then it also depends on this ta, which is daily activity. Well, at noon, maybe I never see a lion, but at dusk and dawn, I might have higher chance, right? So the time of the day will impact my chance of actually seeing one. And then P here, which is the detection probability. That is like, maybe I'm just stupid enough and put my tent in a place where there's trees around and they can walk by and I still don't see them, or I'm this little hill and I see very far, right? So basically, I have a notion of detection too. And I want to learn all these parameters from the data, okay? And as in occupancy, we use environmental covariates for, oh, there's a mistake here. Okay, so the first one is like the density. So we said log of the density is just a linear model with covariates. This should actually be detection, I'm sorry. So this is the logit of detection. It's also environmental covariates. There's a reason why there's no intercept identifiability issues, but basically we use environmental covariates for these, just like we do in occupancy. And then we have some sparse priors on A and B, okay? And then finally, we model these daily activity patterns using piecewise constant function. Okay, this was mass. And you do MCMC. Okay, here's an illustration. Okay, so imagine every row here is a site. So this would be one camera trap. And this camera trap has like some density and some detection probability. And what I show you here in orange is like animals passing that I actually pictured. And then you have like maybe this site 4 here, like it has a very low detection probability. So even though there's animals passing, I never get a picture. And that's just because they have low detection probability. So you see, like to get a lot of these orange things, I need to have a high density and a high detection probability. I need to have both. Finally, we have like these piecewise constant activity functions down here. So we see this is on a day. So at midnight, you never get this species, you only get it like noon, and then you have lower probability to get it. And this is what I want to learn. I want to learn for every place, what is the density, what is the detection, and what is the activity pattern, right? That's what I want to learn for every camera trapping location. Okay, an application, we have 6 species of duikers. These are these tiny, cute antelopes running around. And so for these 6 species of them, we want to kind of to see, okay, can we know something about their distribution, about their time, and what they do? Okay, and here's the results for 2 of those species. So the Grimm's duiker and the yellowback duiker, I think in English. And what is shown here on the right is like our estimate of the time activity function. And what you see is they actually do not really do the same thing. So the green species, the dorsalis, is active in the night. And during the day, you basically get no pictures of this guy, but then during the night again. So these dashed lines show the night. While the Grimm's duiker is like a crepuscular species, so it's most active at dusk and dawn. Okay. We can then also ask what is the habitat they use, and we can make these differential maps. So where I show in dark blue is where there's way more likely to see a Grimm's duiker than the other duiker. Okay, and in green is I see the dorsalis duiker much more likely than the Grimm's duiker. Okay, and you see these species are kind of this species pair is interesting. They kind of use very different habitats. So it seems the dorsalis are here, and the Grimm's duiker prefer to hang around here in the park. And they're also not really active at the same time. So they perfectly partition their niche. They're both small antelopes, eat the same stuff, but they can coexist because they have basically different ecology. Here's another example, right, where there's hardly any difference in habitat use, so there's no real place in space where one of them is much more common than the other. They can still coexist because they have very different feeding patterns. So the Weinsteiger is active during the day, the green is the same as before, dorsal is active during the night. So even though they use basically the habitat the same way, they can coexist because they use very different feeding times. On top here, I show you what we call these overlap coefficients. Other people have developed this before for time. We extended this concept to space, so we can just quantify how much of their distribution, how similar is their distribution actually. And what's kind of cool is like you can make an MDS and a visualization of these overlap coefficients, that's just pairwise distances, to see how these species behave in time and space. So here we have 3 species that are active during the day, 1 that is crepuscular, and 2 somewhat active in the night. They're not exactly doing the same thing, but still. And then here, this is for space. So these guys here prefer savanna, these guys prefer rainforest, and these guys are really ecotone species. They like habitats that are a mix of both. And if you just look at one of these axes, you would say, that's really strange. How can these 2 guys actually coexist if they use the same habitat? Well, because they use different times of the day. And if you do the MDS on the combined thing, you see we no longer have points that are really close together. The closest ones, turns out, are actually these 2, and unsurprisingly, one is dominating and it's very common, and the other one very rare. So that just shows from these camera traps, we learned something about the ecology of the species, and if now one of them is of concern, the people in the park, based on these opportunistic camera traps, could actually say, well, we have to protect this part, or we have to put kind of emphasis here. So this was our goal. Yeah. Okay. Third example. This is the automation thing. So now we talked a lot about camera traps, and camera traps are really cool things. So you put them out, they require little battery. Some of them run for 3 months. You go and collect them 1 year later, they're still intact, you get the data, fantastic. But there's actually also a problem with camera traps, and it's well illustrated in this graph. So here you see in dashed line is all the pictures we've taken in this area, okay? So we are 22 with several million. And in the solid line here is all the camera traps a human actually looked at and tagged. And you see, we just failed to keep up with looking at the images. So the people in the park are taking image after image. But once Thierry quit his PhD, finished his PhD, it has been very slow in actually annotating images. And this really reduces the effectiveness of these tools. So the results I showed you before, they only use basically data until 2016, 17, because that's where we get the data from. We are still collecting, but I could not show you maps of these dikers in 2022 because nobody has identified dikers on these pictures. And why is there this lag? It's not just us. It's like most most projects have this issue. So what is the issue? The issue is that annotation is difficult and still mostly done by experts. So we actually have humans that look at those pictures. Now you would say, well, it's 2024. No, your phone can recognize your cat and your aunt. So basically, why not use AI to actually solve this problem, okay? And it turns out like this is really difficult. So many people have tried, we have also tried, and so it seems obvious, but the accuracy of these tools is just not acceptable for ecological research. And the only papers that you find that claim we solved this, we get 99% accuracy, they usually throw away half of their data, which means throwing away all the rare species where it doesn't work and then have only the common one, and that's just not what we want to do, right? And here's like the confusion matrix when we train AI on our data. And we have millions of pictures. This is a large project, actually. And so these are just some categories. We lumped some into other. So vegetation, we have a 95% chance of identifying an empty picture as empty. Yeah, that's okay. But then you have other things like this guy here. So this is actually buffalo. 40% is recognized as a buffalo, and 37% it's actually think it's an eland, right? And if I show you a picture of a buffalo and an eland, you would not confuse them, okay? And so like there's all these kind of like weird things where you see accuracy is usually below 80% for the majority of the species. And 80% is just not a lot, right? So why is this, right? So the question is, why is training an AI on camera trapping data so difficult? Here's why, okay? There's 3 reasons, I think. First, image quality. That is, we have very interesting images. We have night images, you have day images, rain, sun, everything, a lot of images where the animal is only shown in part. Now that's an eland and you would recognize it. If I only show you the leg, maybe you wouldn't either, right? There's a lot of pictures that are just not in optimal quality for training. Second, the training data is immensely skewed, okay? So here is the number of pictures per species ordered in our data. The most common species has 61,522 human annotated images, where someone said, yes, that's it, that's buffalo. And then here you have this tail where there's actually, say, 20% of all species that we have with a single image. And clearly AI is just not capable of learning those, okay? Third problem is a so-called, what I call the static background problem. Imagine you have camera traps in two locations. One has this nice fancy tree here, and the other one does not, okay? And there's no zebras in the Chinko, so it's just for illustration. But so what happens here is that there's more zebras occurring at this location than at this location, okay? Now what the AI tool is going to do, it's going to say, oh, an image that has the tree here is more likely to have zebra than the other images. And it's true. The AI is doing the right thing, just this is not transferable skills, right? It learns to recognize zebra because there's a tree. If I show it image from another location, it never recognizes zebra because the tree is not there. What that means is actually you have to make sure that you train on a subset, you usually train on a subset of these camera traps to apply the model to another set of camera traps, right? I want to learn with the human, I have data until 2018. I want to use this data to train. And then if my people come in 2024 with the new camera trap, I want to automatically recognize. But what if I trained with this? Oops, and now I want to use it here? It will fail, right? So this is a real problem. All these problems you can address with technical things, but it's difficult. And so I think it's a fact that still accuracy is just not where it should be. So we propose a model to deal with this, okay? And the model is just, let's just do ecology while learning how bad AI is at the same time. Okay, so we basically model here errors. So this is the probability we care about. That's the probability that the AI tells me it's species L, given that the true species is M. And I have two things. I have an error rate and a confusion matrix. And I say, if M equal L, so there's no error, that happens with probability 1 minus making an error. If M is unequal L, so for instance, the truth is lion and it tells me leopard, then that happens because there's an error, epsilon, and then rho is this confusion that says, and if it is an error and it's a lion, you're more likely to say it's a leopard than a zebra. Yeah? So this is the confusion matrix. We say that the image-specific error rates, epsilon, so the logit here, are again a linear function of covariates, such as the AI gives you a lot of hints. Is it good? Is it bad? Like what is the softmax? What is entropy? And also we have other things like, is it a night image or a day image? Maybe there's just more mistakes at night than a day, or actually usually the other way around. And then we have this confusion matrix here, right? Which just says the probability to go from this label to that label. And the model that you then do is something like this. So here's the labeled data by AI. This is the true species. So we say the data should be the true species if there's no error. If there is error, it's different one. This is the covariates that we have here, these betas, and this is R. And then zeta is like what I actually care about. That's ecology. That explains which species is where. And I want to learn this ecology thing here at the same time as these error probabilities from the data. So how does it work? So first of all, you should know that whenever you train AI, you actually not only have one set of data, you actually have multiple. You usually have a true set that you split in like a training data and a validation set, and then you also have a test set. And so this is very important for us because that means we have all these different sets. So this here, for instance, is true set data where I know everything that have not been used in training, and I know the species. So they're gonna give me a lot of information about ecology. Then I have like, this is the regular set. This is, I have AI data, but it was not used in training. This is AI data, but I used it in training. So I actually know the true species, which is super informative about errors and so forth, right? The different partitions of the training data are very informative about different things. And so the model that we have takes all these things into account. Okay, does it work? Okay, I think this is a very interesting slide, I think. So it's some simulations that one of my students did. And we compare here three things. So the first thing is this one here. this line, these kind of like dash dotted line. It's very difficult with that thing to follow, right? So the one that crosses, it starts very low. That is the accuracy we get if you only use manual labels. So what do I consider accuracy here? We have a full data set. Now, the best thing I could do, I could take the full data set, manually label everything without error, and then learn occupancy. This is a simple occupancy model. And then I just ask, what is the correlation between the occupancy that I learn from my full data versus the subset of the data that I have? For instance, if I only use 5% of the data, my occupancy that I learn from the manual 5% is very low because there's a very different results compared to what I get when I have the full data. Now I'm lazy and I would say, okay, but I don't want to label all the data. I want to label only 5%. So how about I use now AI to actually fill in the other 95, right? And then I fill in the other 95 and then I get this curve here, right? Which is interesting. So what is happening here? If you have extremely little data, then the simple AI approach is actually better because you get so much more data. But as you get more data, it's worse than manual labels because it makes so many errors that actually in the end, I'm better in having like 15 or 20% manual is better than 20% manual and the other 80% I use the AI because the AI is so bad, okay? But if we now use an occupancy model that actually learns the AI errors at the same time, you get very interesting things. So if the error rate is 20%, that's 80% accuracy. So my AI gets 80% accurate. Then I get almost the same, almost 100% accuracy if I only label 5% and use AI on the other 95%. So basically AI can be super helpful because I label 5%, I use my AI tool to tell me, okay, now predict the other 95%. But it is only helpful if I account for the errors AI makes. If I don't, I actually don't get good performance, okay? But if I do, I get really good performance, okay? So what that shows is basically AI is helpful, but only if we accept the fact that AI makes a lot of mistakes and we model those mistakes, okay? That's I think the only way to go around this, okay? Now this varies a bit with the actual error rates. So if the error rate is 80%, imagine, that means AI gets 20% correct and 80% is incorrect. We still do reasonably well. So at 50% of the data, I'm still better than manual labeling, okay? So I think that's quite impressive. Okay, almost on time. So here are some conclusions. So for me as a computation biology coming from a different field, working with Thierry has been very interesting because I learned something I didn't expect. I learned that there are still many parts in the world for which we actually have a really bad understanding of biodiversity. And then I see all these fancy papers that model biodiversity and have these fancy maps on the globe. And I just know 1/3 of Africa is just probably not going to be true what these papers say. And we need to change this. And so I kind of embarked on this mission to say, well, let's try to help enabling biodiversity monitoring really worldwide, everywhere, even in the difficult parts in Africa. And I think by using or developing new statistical approaches, we can actually make a big, kind of like a big step forward. And what I think these approaches should do is they should be appropriate for the data that is collected under difficult conditions. There is no standardization, opportunistic sampling, limited data, all these things. We should be interested in these problems because I think these are real world problems. And I think these tools are one big step that bring us closer to, I think, the dream, which is what I call real-time conservation management. This idea that somebody goes out in the field, brings back an SD card of a camera trap, loads into the computer, AI figures out what the species is, automatically I run occupancy or trend models and say, our lions have a problem, right? That's real-time conservation management. We're far from there. But I think it is worth working towards this goal. And yeah, with this, I'd just like to thank a couple of people. So these are 5 PhD students that worked on this. So this is Thierry, then Liam Singer, Madeleine Kadouf, Aime. So Liam and Madeleine worked on Tomcat and Burp. So this trend stuff, and Aime is the one that does AI. Ernest Fotzing is our specialist for environmental covariates, which we use everywhere. And then this is Christoph is a good collaborator from the math department, which is kind of like where we run all our problems through where we get stuck. And then, of course, the rest of my lab and all of the African Parks crew that are actually running the park down there. And if you're interested in the tools, they're all like on our Bitbucket website. Yeah, thank you very much.
Well, thank you very much, Daniel. Do we have questions here?
Thanks, Danielle. I have a question concerning the first topic, the population growth or decline inference. So I wondered about this assumption that you can correctly label the observations. So what you showed is that there was Thierry or you. Well, let's say if your label is it a Jeep driving by or it's a person walking by? Then if it's Thierry or you sitting in the Jeep, it's again not going to be the same thing. So have you thought about this?
Yeah, very much. So I think there's two answers to this. So the first answer is this method is performing well if you respect the design in your data properly. And I just pull up this slide to illustrate it. So what we actually do in the analysis of the real data is we take the road network and I make chunks. Okay, because I say every piece has its own characteristic. There's the soil, it's like how many individuals are around, like maybe lions just don't like this place, but they like this other place. So I don't want to compare this piece of road with this piece of road. And I don't want to compare Thierry spotting lions and me spotting lions. And I don't want to compare wet season and dry season. So we stratify the data in a lot of like these kind of like ways, and then only compare data points that match what we think matters, right? So I say, I think this is a very elegant idea. Personally, I think maybe you disagree, but I think it's elegant because there's always two ways with dealing with these problems. You model it, okay? Or there's three ways. You model it. That's not very fashionable in ecology because models get complicated. You enforce rigorous standardization in the field. I think that's just not possible in these fields. Or three, you find a way around these problems. And this is a way. We stratify the data. So I don't have to make a complicated model, but also I don't ignore the fact that I have these problems. I have standardization. So I do Thierry walked on this road in 2017 with Thierry walked on this road in 22. But I walked this road in 19 and in 23. So I compare my counts of 19 with 23, because if lions increase, I should see more lions and those of 3. And now, of course, there's a trade-off. Like the more kind of like things you have that matter, the less data you kind of get, which is why I advise the people in the field to have this picture kind of in mind. So if you have the choice, there's two rangers, send the same gang to the same road, right? Not flip, right? There's these things. In addition, these methods also kind of like deal with covariates, so that maybe you would just say, we model it differently if you need to. Now, the second answer to this is, well, I would maybe also like to use AI, or maybe humans are just like AI. So clearly the goal would be to have this model work with AI errors in the future. If you work with labeling errors, then the problem is that you have to learn all species at the same time, right? You have to go multi-species. This is currently a single species stuff. And I think there's no reason not to do it. We just haven't done. If you want to bring in label errors and model them specifically, you need to learn the trends of all species at the same time. It's a logical next step, I think.
Yeah, just maybe a more basic question. So for IUCN, they want a prediction of, if I remember correctly, of how long till extinction, right? They have a little bit of a different end goal that they use. And they also do use simple models and try to get people to try to think about them. And so how does your approach fit with the way IUCN is trying to do things? Can they be married somehow?
Well, maybe, but I think a major thing is that under an exponential model, there's never extinction. Right? You just have like an exponential, well, you can say once you have 0.5 individuals, it's probably extinct, but there's no like actual extinction. And I think what often happens in extinctions, which I think is very difficult, is that you get population crash, but then the question is what happens when there's really few? And there's different things that can happen. So I come from population genetics where people talk about extinction vortex, and then very often you get like a very sudden crash. And I think actually predicting that sudden crash is very difficult. So I would give the question back. So is actually time to extinction, is that really the right question to ask? I think for many reasons, first of all, I think it's very difficult to predict, okay? So you need a lot of data, and by the time you figured it out, extinction happened maybe in a like sarcastic way. But the second thing is also, we want to prevent extinction, no? So if I'm IUCN and say, well, it takes 20 years to extinction, and this species takes 10 years to extinction. what am I now going to do? Am I just going to focus on the 10-year extinction so that in 10 years I have the same problem in the next species? I think just time to extinction, I know it's just my opinion, but I think time to extinction is maybe not even the appropriate thing. I think if I'm a manager of an area, my goal is that my species don't decline. And every decline is kind of bad. And of course, I need to prioritize. So I want to basically know what's happening right now and how can I change it. And I think that's what we focus on. IUCN has different goals and their goals are very valid. But I think like marrying these things, I'm not sure. This is a short-term thing. Because as you go for long-term, there's lots of other things you can ask. Like, well, how sure do you need to be in this, right? Down here, it's a very flat curve. So just a little bit noise up here will make suddenly a big difference when you predict. I just think it's a very difficult problem. It's a different problem, right? I don't know if this answers this, but it's just like, it's not something I try to forecast. I think it's very difficult to forecast. I'd rather just say, what I'm interested in is what people do in the field. They see this species declines. You want to do something. What you want to get is immediate feedback if what you do makes sense. Right? And that means after 2 years, I want to know, should I change my strategy or should I continue doing this? So what I want is or what I care about is the people that say, I do something, now let's see. But as quickly as possible, we want to get kind of like feedback. The 20-year prediction is anyway wrong because it is under the assumption we do nothing. But we will do something, right? So this prediction is very difficult and not something we try to do. Sorry.
I have a question about the third part of your talk about correcting AI mistakes. I'm wondering how generally applicable is the concept, and also if you have something published on the topic where I can read more.
Yeah, so I think it's very it's broadly applicable, but you have to change all methods, okay? But I think it's okay because I don't know if you're familiar with DAG. So this is just a directed acyclic graph illustration for probabilistic model, okay? But what's really nice is what you see is like this theta is basically the model you already have. In order to apply this, you have to do two things. You have to stack this below, right? This model, but you can copy it to your model, I think. The second thing, your model has to be multi-species, yeah? And that makes immediate sense. If my data can be either a lion or a zebra, I need to work with lions and zebra at the same time, right? So basically, you can directly copy our mass and actually compute the code into your model if your model is multi-species. Okay? If it's not yet multi-species, you first have to make it multi-species. Now, in the occupancy model we use here, we make a very simple multi-species thing. We call this, well, how did Aire call this? He had a nice name for this. But it's just like, there's fancy multi-species models. They model interactions between species and I don't know what. But you can have a much simpler approach. It just says, if I take, so that's what we do in occupancy. If you have an image, what's the probability this image is from this or that species? We just have relative densities. And that's all you need to go multi-species in occupancy in the simplest form. Yeah, so you have to go multi-species. If you are, you can just copy it. So I think it's probably applicable. Yeah. Now you ask whether it's published. No, Aire is just writing up the manuscript. I hope in 2 months or so this is published. Yeah, or at least on bioarchive.
So maybe one last question, and then yeah, I mean there's two, but what I suggest is anyhow we'll have lunch afterwards. So you can talk with Daniel during lunch. There's a table reserved for everybody, although you have to pay for food. And next, there's a room which is booked if you want to have a one-to-one discussion with Daniel. I know Luca was interested. If some of you are interested, please, yeah, talk to me and we'll arrange that. So last question, and then should we? Oh, okay, it's you. All right. Okay. All right. Cool. Go ahead.
Quickly, I was just curious. So there's this biodiversity market hype and a lot of startups collecting data and channeling donations to people on the ground through blockchain and AI images and stuff like this. I wonder if you've been approached by any of these startups, or what do you think about this market and the potential use of such images to do real conservation
Okay, well, that's a tough question. So I think I haven't been approached by people. Maybe that will change. I think the market. Well, we have a lot of discussions about this, okay, with the people from the park, and I'm gonna probably make a bold statement, but like I think a lot of the current ways of monetizing these things have inherent flaws. Maybe it's not easy to find something that does not have flaws. I cannot really present you the perfect thing, but if as soon as we start to funnel money based on indicators, we just have to be very clear about what kind of incentives that creates. I think a good example from the past is endemics. So the more endemics you have, the more money you get, just means that every species is chopped up into 500 subspecies so that everyone gets their endemic, right? And we can think about other things. So basically say the carbon stock has this major flaw, which is basically just as a forest gets money if it stocks carbon and is threatened. So if we work in an area like the Chinko, you don't get money because, well, nobody wants to cut it down. So in order to get money, I need to build a road and have somebody chopping down the forest around me because then I get money. That's really not the incentive you want to have if you want to protect forests. So I think almost all of these simple things, they create weird incentives for people to act. And I think whenever we propose something, we have to be very careful about this because we see these kind of weird things happening on the ground where suddenly you say, yes, the forest next to me is cut down because now I get money, which is really opposite to what you want to achieve. So personally, what I think is actually probably if you just kind of like have, or like you finance areas that are in an almost natural state and you give preference for this, I think it's very hard to have very wrong, or it's more difficult to get wrong incentives, right? Somehow, if you get money for leaving the stuff in a wild state, you don't do much wrong, I would argue. Now, that's maybe my opinion, and people have different opinions, but I just think this market is a very difficult thing. And I have not been involved in this research. We just discussed, so this park that we work with is in the African Parks Network, and they have now more than 30 parks, I think, across Africa, and they try to get money. And you see have exactly these discussions. So the manager of 30 parks come, how can we get more of carbon stock? How do we have to do it? And these people will take action so they maximize the money they get. So if you put out an index, it has real world consequences of what people are doing. And a lot of these indices we have right now, I think, have been thought through from one aspect, but nobody has really thought about what kind of bad incentives does that create? And I just think we have to be extremely careful. This is, yeah.
Well, thanks. Let's thank again Daniel, who's been very good at handling the noise, which was super annoying. Yeah, thanks again, Daniel.
You're welcome.